you for joining us tonight. We greatly appreciate your interest in this clinical trial because the potential it offers us for a new scar-free preventive option to reduce our BCC burden is absolutely incredible. In my opinion, clinical trial participants are true heroes to the Gorlin syndrome community. People who do this give their time, their effort to advance the cause and potentially improve all of our quality of lives. So thank you for being here. Let's start with a few housekeeping items. This webinar is being recorded and will be available on our website and social media channels in the next few days. Everyone is muted during the webinar, and you may choose whether you have your camera on or not. Prior to the webinar, we offered a window of time for people to send questions, and we'll respond to these at the end. Once we're through them, if there's time, we'll open it up for attendees. But I ask that you remember that we can only take clinical trial-related questions during this time. That is the sole purpose of this webinar. If you have other questions about Gorlin syndrome or about issues you're having, or if we're unable to get to your clinical trial-related question, please don't hesitate to email us at info@gorlinsyndrome.org. I'm going to share my screen now, and we will get started. Come on, share screen. Time to work. I mean, slideshow, time to go. Well, it doesn't want to happen. So we will just go with it like this. Sorry about that. Can everybody see the whole screen adequately? So tonight we're here to talk about the clinical trial of SGT 610-01. This is a trial being put on by Sol-Gel Pharmaceuticals. Their mission is to harness dermatologic innovation for patients seeking transformative outcomes. And as a reminder, our mission, that of the Gorlin Syndrome Alliance, is to thoughtfully support, comprehensively educate, and aggressively seek the best treatments and ultimately a cure for those affected with Gorlin syndrome. Our main speaker tonight is our friend. Dr. Epstein has been part of the Gorlin syndrome community for as long or longer than many of us have been on this earth. He has been studying patients, and well, it's hard for me to really express all he means to us. And I just hope, here we go. I just am very grateful for Dr. Epstein and all he's done with us and for us and all he continues to do in these efforts. So now we'll begin. Thank you, Dr. Epstein, for being here tonight. And I'll turn it over to you.
Okay, let's get that slide off. Go to the next slide. Thank you. I would like to echo what Julie said about people who participate in trials or even think about participating in trials and being heroes for the whole community. It may be for your grandchildren, it may be for your children benefit, and hopefully it will be for your own benefit as well. I've asked people to do various things over the last 50 years in terms of helping my research, helping the research, and I have said the chances are it won't work. But if we don't do it, the chances are 100% it won't work. And actually, I'm proud to say that in the 50 years since the first paper appeared with my name as a co-author, there have actually been significant improvements in what we can do for patients with Gorlin syndrome. So I am confident in what I say, and hopefully the confidence will not be misplaced. So want to talk about Petitagib today. This is incidentally a beautiful late spring afternoon in San Francisco area, and I appreciate people giving up time outside in this beautiful weather, or even just looking out the window. But this is the topical version of a drug that will work in the same, that works in the same manner as does Aravage, also known as Odomzo, or excuse me, as Vismodegib, or Odomzo, also known as Sunitigib. And as probably most of you know, in 1995, our lab, collaborators, competitors, a whole huge group of people in the global village identified the gene that's the site of the typo, the mutation in the DNA, typo in the DNA that causes Gorlin syndrome, at least in most patients. And that's called the patch1 gene. And that was, for that time, a great triumph. Now, not as interesting as it was in 1995, but things get better. And really, the history of medicine in the last 50 years has been of it getting much, much better. And we, two friends and I, were sitting around at Stanford 14 years ago saying, okay, Genentech has brought on the market this fabulous drug called Aravage, which I tease as a mistake. It should have been Irv's Edge, but they deny that, but okay. And you could have a patient with Gorlin syndrome who has 100 basal cell carcinomas. You give them one pill a day for a year, and they can be all gone. 90% of patients, they're all gone. And if you do a biopsy at the places where the basal cell carcinoma used to be, you see nothing. It just looks like normal skin. So that is stunning. Not chemotherapy. It's really attacking the result of the mutation and replacing the function of the defective gene product. But even though it's that terrific, when you stop the drug, of course, most of the lesions come back, even if they looked absolutely normal under the microscope. So why would anybody stop it? Well, most of you probably know that this class of drugs, Vismodegib, Sunitigib, Odomzo, Aravage, and indeed Petitagib, when given orally, all these drugs when given orally are different medications, different molecules, different active molecules, but they still attack the exact same target. And they all have the same efficacy by mouth and all the same side effects, which include especially hair loss, taste loss, and muscle cramps. So we were saying in the bright sunlight, back to the three of us at Stanford 14 years ago, there must be a better way of doing this. That maybe somehow we could find something that was not quite, perhaps not quite as effective as Aravage, but nonetheless, it had fewer of the side effects and would be more easy for people to tolerate long-term, because of course, people with Gorlin syndrome are developing new BCCs all the time. It's not one and done by any means. If you walk out of the Mohs surgeon's office with no BCCs, you're pretty sure you're going to get some more within not too long. So how could we make something that could be used for life and have at least some of the efficacy of Aravage? And being that 2 out of the 3 of us are dermatologists, well, duh, the answer was what dermatologists do, which is mostly, at least in the past, to write prescriptions for people to take to the pharmacy and get a topical agent, a cream, an ointment, a gel, something to put on the skin in the area where there's a problem, and hopefully it will reverse the problem, be that eczema or psoriasis or whatever. So why don't we take something like Aravage and put it into a cream form and have people put it on the skin in hopes that we might treat existing tumors, or at least help reduce the number of new tumors that develop. So we had to start a company because it's far too much money to do this. In fact, it's an obscene amount of money to do this kind of work. And you can't ever get enough of this from an NIH grant. But the NIH grant was enough to get us the understanding about the patch gene and to understand how it worked. So we decided to make a topical drug And we wanted to make a quote unquote Goldilocks drug. Next slide. What do I mean by a Goldilocks drug? I mean a drug that when you put it on the skin locally, will have enough of the active ingredient that gets into the skin to have a beneficial effect on the area where you're putting it. It will be efficacy locally, but not so much get it in that it spills over to the circulation and causes the distant side effects like hair loss, taste loss, and muscle cramps that plague people who are taking these oral inhibitors. So next slide. What we did is we outlicensed a drug, a molecule that worked about as well orally as the oral hedgehog inhibitors, Aravig and Adomzo, and we made it up into a gel form. We did phase 1 trials, which showed that it seems to be safe and maybe has a little bit of efficacy. We did a phase 2 trial that showed that it had pretty good efficacy, what we could see in a small number of people. And then we did a phase 3 trial in our company, which was called Pelifon. And the phase 3 trial is a trial where you put in all the bells and whistles that will allow you to convincingly demonstrate safety, first of all, and efficacy. And if you do it well enough, the FDA will most likely approve that you were able to bring it to patients. You can put it on the market. So that was the phase 3 trial that we did starting in about 2019. We included people who were diagnosed as having Gorlin syndrome with many BCCs, jaw cysts, pits of the palms and soles, and other clinical criteria. So this was just people who by clinical criteria had Gorlin syndrome. We enrolled 174 participants at 44 different sites in 10 countries on 2 continents, aka North America and Europe. Of those, it was randomized to 50-50, 1-to-1, of applying either the active gel twice a day all over the face, or the other 87 applied the placebo, the gel in which the active ingredient was suspended, but no active drug. And I should say at this point that it's very important to have information from both those who are applying active drug and those who are applying placebo, especially if it's like this trial, which is trying to look at prevention. If you're looking at something going away, that's pretty easy for all of us to recognize. But to understand something that didn't happen is very difficult. So you need a placebo to see what would have happened in these patients as closely matched to the others as possible, as to the others who are putting on the active, to see how many they got and see if you had the people who were putting on the active had fewer during the course of the year. And of course, we hope that the endpoint of the development of new BCCs that could be seen by the naked eye, really, and by photographs, that would be eventually this spill over into fewer surgeries, less discomfort, and less scarring. So there was good news and disappointing news. At the end of this, it's immensely expensive. Lots of people doing lots of effort, even on through COVID. The good news is that there were really no significant number of side effects in the active as compared to the placebo. A couple of patients had a little bit of irritation where they put it on. They stopped it for a day or two and they were able to start it up again without problems. And there was just as much of that in the placebo as in the active. So none of the hair loss, taste loss, and muscle cramps. Although if you put it, some people were putting it on the front part of the scalp instead of just leaving an area where they didn't put it on between the scalp and where they put it on the face. And they did get some hair loss, which is a known, which was not surprising. So no significant number of side effects, no hair loss, taste loss, muscle cramps, and the rest of the problems. And those applying the active drug, if you included all 174 who put it on at least once, just once, we found that there were 30% fewer new BCCs on the face during the year that they applied. Okay, so the good news is no significant number of side effects and some 30% fewer new facial BCCs during the year they applied. excuse me if I cough and if I'm a little scratchy. I tested positive for COVID 2 weeks ago tomorrow, but feel just fine, except for that little lingering cough. The disappointing news, of course, is there was a lot of overlap, and the efficacy seen could have been random. So the FDA is insistent on a lot of statistical analysis. And what is the statistics? What is it trying to tell you? Well, I use the analogy, and it's not mine, of course. If you take a coin and you flip it 5 times in a row and it comes up heads every time, you'll say there's something wrong here. It's not coming up. You expect it to come up 2.5%, 2.5 times heads and 2.5 times tails. So there's something wrong. If you flip it 1,000 times, then having during that 1,000 times a run of 5 in a row, that's not so surprising. You know that that can occur. So statistics tries to tell you, is this just a fluke or is it something that's real? And the statistical analysis of the all-comers, the 174 patients, the 30% reduction, was such that it argued that this could have been a fluke. Now, anything could be a fluke, even 5 times in a row that you flip a coin, it comes up heads. But still, you want to be 95% certain, 98% certain, something like that, that what you're seeing is a real finding and is reproducible. So that was disappointing that it was not good enough for the FDA to say, yeah, we like the fact there's no side effects and we see it's 30% of efficacy and we'll let you market it. They did not say that. They said, go back and try again. And what we did is we looked at and we analyzed more carefully the results of the 110 participants who actually had a patch mutation. So as I said earlier, we did not require people enrolled to have a significant, to even be tested for a mutation in the genes that we know can underlie Gorlin syndrome. We offered it as the choice of the patients. 130 of the 174 said, yeah, test my blood. And of those, 110 out of 130, 85% of them did have a mutation in patch, a typo in the DNA sequence that we figured, yes, that is the cause of their problem. And then we looked at people who had enough facial basal cell carcinomas at the start. What do I mean by enough? What was the point of measuring, of numbering how many they had at the beginning? The point was we found out afterward, but it's not surprising, that those who had more than the median, more than, we lined up all the patients who had of the 110, and we saw that the number they had at baseline was predictive of how many they would have in the next year. In other words, people who had a lot at the baseline were likely to have lots at the end, by the end of the year, and people who had very few were likely to have few. And so it's not that we expect the cream to work better or worse in people who have many as compared to people who have fewer, but or who develop many versus people who develop fewer, have a fast tempo versus slow tempo in new development. But for the purposes of trial and getting a statistical analysis without enrolling 1,000 people, we needed people who were going to have a lot of tumors. So if we looked at only of those 110 who had the patch mutation, we just looked at the 50% who had the people had more than the 50% who didn't have it. Just looked at those who had at least 11 on the face at the start, and these could be large or they could be very small. If we looked at those, then it was a 50% reduction in development of new tumors. We just had more new tumors in the placebo to work with. And the results give a p-value of less than 0.01, which is definitely even better than a holy grail. It means, roughly speaking, and I am by no means a statistician. I have a book by my bedside that I read when I can't fall asleep, and I've never gotten to page 2. It's called Biostatistics for Dummies. That puts me right to sleep. But it means approximately, gives you 99% likelihood that if you repeat it, you'll get the same result time after time. So we talked to a number of people, a number of companies, and asked them, how about giving us money to do another trial? And basically, there was a lot of interest, but at the end, we didn't get anybody. And what we got was Sol-Gel, which is a very good company in Israel that is devoted solely to making small molecules that can be applied topically, applied to the skin, to cause a reduction in whatever disease that's being treated, acne or psoriasis or whatever. And they bought it. And so it is now their product completely. And it's not my company. I am a consultant. I get paid to help them set up trial sites and help them recruit patients. And that's what I'm doing. So next slide. So as I said, we sold the project to Sol-Gel, and they've opened a trial of only people who not only have a clinical diagnosis of Gorlin syndrome, but also have a detectable patch mutation, which appears to abrogate the effect of the protein that's encoded by patch 1, and at least 10 on the face at the start of the trial. And we expect, and if our statistics are right, it's 99% likely, but you never know. The results will mimic those seen before, with 50% fewer new BCCs, no side effects, like those seen with oral drugs. It isn't that the drug is magic. What's magic and different is the way it's being applied to the skin and not to the body, by mouth. And we anticipate that if this is as successful as we think it will be, the FDA will allow Sol-Gel to allow the drug to be used by Gorlin patients. So the goal is that Gorlin syndrome patients who apply it regularly will have a 50% overall reduction, on average reduction in development in new BCCs during the course of the year. And hopefully that will translate eventually to a meaningful reduction in the number of surgeries they will require. I personally, and I will tell you, I have absolutely no data to show that that's true. And so many of the things that in the last 50 years in science I have thought made sense and would be true turned out to be false. And this may be false too, but I think that with longer and longer use, we'll basically be treating tumors in the very first cell that develops, and it will be better than 50%. But I don't know that that's going to happen. I know it's not going to happen in a year, but I'm hoping that there'll be a meaningful reduction in the number of surgeries and eventually the number of scarring that the community develops. Next slide.
Irvin, I have to make a quick switch here. I've realized that I'm sharing the, never mind. Just so everybody understands, we've had multiple derivations of this presentation, and
I noticed
this
it,
one,
I thought we'd correct it.
yeah,
But I didn't have to point that out.
this one is, we're just going to keep going with this one. So pardon the interruption there. Let me get us back to the right.
Let's go on from that.
Yeah, hang on. I know it's not your fave.
Well, it is my favorite because all the other pictures make me look dead, and I don't like that. I much prefer this one, which at least to me makes me look alive. So if you want more,
keep going.
If you want more specific information, you can go to the website that's managed by the federal government called clinicaltrials.gov. And that is a website that has basically all the recently concluded, maybe since it was started some years ago, 10 years ago, something like that, all the clinical trials that have been registered. How do they get people to register them? Because if it fails, you might not want that known. And the answer is you can't really publish it in a first-class journal if you didn't register it ahead of time. So they've been pretty good about managing to get most all the clinical trials onto that website. And you can see 3 or 4 trials we did under Gorlin syndrome. If you look up Gorlin syndrome, if you look up Gorlin syndrome and Sol-Gel, or maybe just Sol-Gel, you'll go directly to this trial. And it's called the efficacy and safety of patidogenic 2% gel for preventing basal cell carcinomas on the face of adults with Gorlin syndrome. So it's got a more tongue twister title, but you can see that. Next slide. And brief summary, as I really implied, the aim is to find out if we can reduce the number of new BCCs that patients with Gorlin syndrome develop on the face for the next year. I should say that why the face? And there were really three reasons. We don't think that it will work only on the face. We think it will have some efficacy elsewhere, whether it's greater or lesser, we don't know, but we think it will have, but we don't know. But if three reasons for choosing the face are: number one, that's the site that most people are most concerned about in terms of the discomfort of having the surgery, in terms of scarring, and in terms of the bandages. So that seems reasonable. The second is that because it's on the face and you can look in the mirror, you can put it on yourself. You don't need a partner to help you put it on like you would on the scalp or the back. And number three, it has landmarks on it. So we're taking careful pictures at the first time and the second time and the end of the trial and reviewing that. And we have fairly good confidence based on our first trial that if you go back to a place that's, let's say, 1.5 centimeters to the left of the left eye, at the beginning and at the end, you can go to the same place. So that's why the face. If you look at the scalp, unless you shave it, and even then, it's very difficult if you don't see any landmarks like the eye or the nose or the mouth to know if you're in the same place as you were a year ago. So participants will either apply Patinative gel, the same gel we used in the previous trial, or a vehicle gel, the same vehicle gel that was used in our first phase 3 trial, to the face twice a day for 12 months. And basically the endpoint is comparing the number of new BCCs on the face between those who use the active gel and those who use the placebo. And I can't emphasize again enough that each of those, the data from each of those is equally important. The use of vehicle or the use of active gel. Next slide. So we're doing it again in Europe and the United States, randomized. It's double-blinded, which means that nobody associated with the trial, not the participants, not the sites, not Sol-Gel, not me, not the pharmacists who dispense it. Only the research pharmacists will know who got which. So if there's some emergency and you need to break it, it can be broken even on a Sunday, hopefully. Applied twice daily to the face of adult participants with Gorlin syndrome. Again, has 1-to-1 randomization between either the active or the placebo gel, and all BCCs at baseline and all new ones will be developed during the study will be photographed and tracked as carefully as we possibly can. Next slide. Eligibility. So the subject must be at least 18 years old at the screening visit. Why not younger people? Wouldn't it be good to treat kids who are diagnosed with Gorlin syndrome before they even have their first basal cell carcinoma? We agree completely. The problem is that if you take this drug by mouth, emphasize by mouth, and you haven't reached full development size, your long bones stop growing. And this happens in mice, and it happens in people where they've been given the oral drug for non-skin cancer purposes. They just stop growing. So we figure by 18 years of age, most people would have reached their adult height within a few inches. And if they have lots of basal cell carcinomas, it would be good to use it for them. If it works and it can be marketed and it's safe, then we'll start looking at 15-year-olds and 12-year-olds and hopefully get down to very young toddlers who before they ever develop tumors. And the subject must be confirmed to have a patch mutation, so we have a chance of looking at 50% inhibition as opposed to the 30% inhibition of all comers who are clinically diagnosed. Got to have at least 10 on the face so that even with the patch mutation, you're more likely to have more in the next year. And so there'll be something for us to see a lower number. And the FDA understands that this is not something, as I said before, that means that we believe it'll only work in people with 10 or more. We believe it'll work with people who have none eventually. But that's what you need in order to see something that's statistically significant. And of course, you have to not use other stuff on the face. Yeah, can't use Odomzo or Araveg or other things orally that go to the skin. But Hippocrates or somebody even older than I am said, you've got to, for the first thing, for a doctor, first do no harm. And certainly if you have a lesion, a basal cell carcinoma that's growing next to the eye, and your dermatologist says, I don't think that's safe to leave it there, then he or she takes it out. But we want to make sure it's been caught in a picture and we know clearly that it's been taken out. So that's why these criteria. Next slide. Now, you can't have used a topical treatment like this in recent days, not any of the topical treatments which are believed to prevent basal cell carcinomas. Same goes for systemic therapies. And I think it's at least 3 months after you stop something. You can't use it if you're known to be sensitive to any of the ingredients. How do you know if you're sensitive to any ingredients? You probably wouldn't. But fortunately, at least in the patients we've used it on so far, there's not been any significant hypersensitivity. You don't have any uncontrolled systemic disease. You haven't been treated for invasive cancer in the past 5 years. And if you used one of the oral hedgehog inhibitors and stopped because it failed, it was basically reasons that were unfavorable side effects. It wasn't that it didn't work, because we think that if Odomzo or Sunitinib, Odomzo, or Araveg, Vismodegib didn't work, ours isn't going to work either, because they're the same kind. They're not the same molecule, but they're the same type of molecule that we're using. And so no point in your getting involved with that. Next slide. As said, I needed a glass of water, drink of water. The outcome that we're measuring is the number of new basal cell carcinomas on the face at month 12, confirmed through careful examination in person and through pictures. And it's the people who applied the active versus the people who applied the vehicle. Next slide.